So my name is Songi. I'm a professor in the Department of Chemistry and Biochemistry, as well as the Department of Chemical Engineering at the University of California, Santa Barbara. I consider myself both a biophysical chemist as well as a spectroscopist. So I'm very much at the basic science of developing methods, and specifically characterization methods to look more closely into proteins and how proteins interact. We are really interested in how tau, from its soluble form, forms fibrillar aggregates as found under neurodegenerative conditions. But my study focuses on the molecular mechanism, on the biophysical molecular mechanisms of really at the atomistic and protein level, what shape changes are occurring, what is starting this process, what is driving this process. So when we discover those factors, it doesn't mean that those factors are necessarily the factors that will occur in the brain or the human brain, but it will give us hints at how this process can happen, and gives us clues into how we can improve really the biomedical studies of these important proteins that are diagnostic of so many devastating neurodegenerative disease conditions. In my talk here will be the shape changes and transformation of tau as it goes from a soluble form to fibrillar plaque reminiscent of pathological fibrils. Here is a key finding. The plaques of this protein tau in the brain, its location, morphology, has been known for a long time to be diagnostic of these devastating neurodegenerative diseases. What also has been known is that you can take then those brain-derived material and can replicate some of this behavior in animal models, and more recently even in cell models, in a prion-like behavior. That part has been studied and pioneered by many, including researchers here at UPenn. All of these in vivo studies is important and wonderful, but in order to really get at the shape change and structure and mechanism, we need biophysical tool to study the structure. What that means is we need to be able to replicate this disease properties of propagation in the test tube. That has been very difficult. We discovered what we call a three-player game, where you need that seeding material that is brain-derived, or at least from cell-derived, tau, which is the one that's undergoing shape changes, and a cofactor. That is our discovery, that you need a cofactor that mildly interacts with the tau, lowers this barrier, and now when you add this a human-derived material, for example, what is called as a seed, the reaction takes off. So we were able to replicate the aggregation of tau seeded by brain-derived material. That in and of itself isn't a breakthrough yet, but it can lead to breakthrough in that then the structure of this resulting fibril can now be studied in much greater detail than is possible with in vivo systems.